Huntington's disease is caused by having a long CAG repeat in the HTT gene. Scientists have found that the CAG repeats continue to get longer in the brains and liver of people with HD as the disease progresses, called repeat expansion. Scientists are looking for different methods to stop CAG repeat expansion. A group in Boston led by Dr. Jong-Min Lee recently applied cutting-edge technology to try to change the letters of the CAG repeat in cells grown in a dish, and in mice that model HD. The genetic code of every living organism is made up of 4 letters: C, A, G, and T. They are combined in different ways to make every gene in our body, much like the letters on each page of a recipe book. For example, the DNA sequence CAG tells the body to make glutamine. This is one of the amino acids that combine in different ways to build the proteins in our bodies. Interestingly, another code, CAA, provides the same instruction to the body and also codes for glutamine. In most people, the sequence of expanded CAG repeats is interrupted by a CAA sequence. When the CAG repeat is interrupted by CAA, it does not change the protein produced, since both CAG and CAA code for glutamine. Scientists have found that there is a difference in the age of onset of HD depending on the presence or absence of a CAA interruption. The age of onset is predicted to be a few years earlier when there are no CAA sequences that interrupt the CAG repeat tract sequence. When there are extra CAA sequences present, the predicted result is a delayed age of onset. So there may be something special about those CAA letters that interrupt the repeated CAG letters. Dr. Lee's group have used a new technology called base editing, which is a special type of CRISPR system. Base editing can change a single genetic letter, allowing CAG to be changed to CAA, which would create more interruptions in the CAG sequence. CRISPR technology has recently been used to develop medicines that have received regulatory agency approval, such as for sickle cell disease. As this is a new technology, the Lee group first performed base editing on cells growing in dishes. The scientists tried different combinations of base editing ingredients to test what happened in cells that have a longer CAG repeat. Excitingly, when cells were treated with some of the base editing ingredients, up to 50% of the cells in the dish had formed CAA interruptions within the CAG repeat sequence. However, scientists cannot precisely control where these interruptions are added in the CAG repeats. This is quite encouraging, as this technology could help delay the onset of HD, but getting all the ingredients to make base editing work into the brain so people with HD is a big task. This is similar to the hurdles facing CRISPR therapeutics for any brain disorder. More experiments need to be done before base editing can be used in humans, but we're excited to hear more about this promising work in the future.